Ever wondered how the mind-boggling complexity of our bodies translates into physical reality from a simple DNA molecule? Well, buckle up. Because today we delve deep into the labyrinth of life's code, through the process known as transcription. Picture, if you will, the double helix structure of DNA, with its matching pairs of nucleotides, the building blocks of life. Transcription begins in this microscopic world, when an enzyme named RNA polymerase latches onto the DNA molecule at a specific point, the promoter. RNA polymerase doesn't just jump onto any random spot on the DNA, it has a precise starting point. This precision ensures that the correct gene gets transcribed. Once RNA polymerase binds to the promoter, it starts to unwind the DNA, creating a bubble-like structure. This bubble is where the magic happens. Once the DNA unwinds, one of the DNA strands, known as the template strand, is ready for transcription. As RNA polymerase moves along this template strand, it reads the nucleotides and starts to build a strand of messenger RNA, or mRNA, from free nucleotides floating around in the nucleus. Here's where it gets fascinating. The RNA polymerase doesn't just copy the DNA. Instead, it mirrors the DNA's code. For every adenine on the DNA, it adds a uracil to the mRNA. And for every cytosine, it adds a guanine. This way, it creates a complementary sequence to the DNA but with uracil replacing thymine. Once the RNA polymerase reaches a sequence of DNA called the terminator, it knows it's time to stop. It releases the DNA and the newly formed mRNA. The mRNA then leaves the nucleus and enters the cytoplasm, carrying the genetic information from the DNA to the machinery that will translate it into proteins. So to recap, transcription is a meticulous process that begins when RNA polymerase binds to the DNA's promoter. It unwinds the DNA, creates a transcription bubble, and begins to build a complementary mRNA strand. The process ends when RNA polymerase reaches the terminator, and the mRNA strand, now a messenger carrying the DNA's instructions, leaves the nucleus. This process of transcription is not just a biological phenomenon, it's a symphony of life, a dance of molecules that happens countless times every second in every cell of our bodies. It's a testament to the intricate beauty of life, where a simple code can give rise to the incredible complexity and diversity we see in the living world. Remember, understanding transcription is not just about learning the steps. It's about appreciating the wonder of life's inner workings, the marvel of how a simple molecule can dictate the workings of an entire organism. It's about marveling at the miracle of life itself.